This video is going to be the first video in a series on eukaryotic and prokaryotic cells. So like always, we're going to divide this really massive topic into kind of small bite-sized pieces. So we're going to do a quick review of what a cell is, and then we're going to talk about prokaryotic cells, just a general overview, and then how we describe the shape and arrangement of bacterial cells, because in this microbiology course, we do spend a lot of time talking about bacteria, and that's what we're going to be seeing in lab as well. So cells are the basic unit of life. A cell is able to carry out all the necessary reactions and functions to keep something alive. So some organisms are made up of one single cell. We say that those organisms are unicellular. Other organisms, like humans, are multicellular. We're made up of lots of different cells. Humans, we have billions of cells. Bacteria, just one single cell. We can further classify cells into one of two categories, prokaryotes and eukaryotes. So depending on how you're speaking about cells, you might say a bacteria is a prokaryote. Or you could say this picture on the slide is a prokaryotic cell. Okay, so again, it's just about grammar. So prokaryotes are typically smaller and structurally more simple than a eukaryotic cell. And we're going to see a picture of one in just a second. But if you take a look at this bacterial cell here, because bacteria are prokaryotes, you'll notice there's quite a lot going on the outside of the cell. There's a lot of accessory structures. So those are things that some bacteria have. But if we focus our attention on the inside, okay, you're going to see this dense region where all the DNA is. See a lot of these little dots. We're going to talk about what those are in a future video. But notice the inside of a prokaryotic cell is really just space with a few things floating around inside. If we compare this to a eukaryotic cell, look at how more structurally complex this eukaryotic cell is. It has a lot of internal organization. And you're going to learn in future videos that we refer to these different components as organelles. Okay, so just like the human body, we have a lot of organs inside of us, right? They have their own shape, their own job, right, to help keep us alive. That's what an organelle is. It's a tiny organ that has a certain role within a eukaryotic cell. Now, prokaryotes do not have organelles, okay, and they also don't have a nucleus. The nucleus is the specific region of a eukaryotic cell where the DNA is held. Okay, so just real quick, kind of big picture, prokaryotes tend to be smaller and more structurally simple than a eukaryotic cell. So let's kind of focus our attention on prokaryotic cells for now. As I mentioned, prokaryotic organisms are made up of one single cell. In all of E. coli's glory, it is one single cell. So again, the science term for that is unicellular. As I pointed out on the previous slide, there are no real internal structure, no internal organization. Everything that exists inside of a prokaryotic cell, it's just all in there in that one space, kind of mingled, hanging out together. And as I said, no nucleus. The DNA is not separate from everything else that's inside the cell. And again, they're all just kind of commingling. So before we zoom in on these individual components and really get into the details, let's first talk about how we describe the shape of bacterial cells, because we're going to use this knowledge in both lecture and lab. So when we're in microbiology, you're rarely going to hear me say shape, because we're going to be building our science vocabulary, and the science word for shape is morphology. So bacterial morphologies are the different bacterial shapes you're going to observe. So if you take a look at this table, notice one morphology of a bacterial cell is coccus. So you would say a single cell is coccus if it is round. And now cells are 3D, so it's more of a sphere. If we're looking at a bunch of spherical cells, you would use the term cocci. Okay, that's plural. If the bacteria is more of like a rod-shaped cell, then we say bacillus for singular or bacilli for plural. One that we will talk about later in this semester is Vibrio, and those are comma-shaped cells like you see down here. And then we're never going to really talk about Coccobacillus, so don't worry about that one for now. And then we're going to have some spiral-shaped bacteria. And we're mainly going to talk about spirillum, but spirochetes are also spiral-shaped cells. And if you compare the picture on the PowerPoint slide, you'll notice that spirochetes tend to be thinner, a little bit more wiry and coily than a spirillum. Okay, so they're both spiral. It's just a matter of how thick is the cell and how many coils make up that spiral. So the four we're mainly going to talk about in class: coccus, bacillus, spirochete, and spirillum. So again, these are bacterial morphologies. So when I ask you about the morphology, just look at the overall shape. And you can really do this by just looking at a single cell. But oftentimes, cells don't just hang out by themselves, okay? As bacteria divide, sometimes they remain in contact with other cells. So when some organisms, when the cell divides, they do remain as separate entities. We refer to that arrangement as single. Okay, so if you were looking through a microscope, you would just see a bunch of single circular cells, for instance. So we would describe the arrangement as single and the morphology as coccus. We might instead look through the microscope and see a bunch of rod-shaped cells hanging out by themselves. That arrangement, again, would be single, but we would use the term bacillus to refer to that rod shape. What about other arrangements? What if some of the cells like to stay in contact with other cells? So if you look through the microscope and you see cells hanging out in pairs, we refer to that as diplo. If you see them in fours, that's going to be tetrads. If you see cells arranged in groups or clusters, then we use the term staphylo. And then if you see cells arranged in chains or lines, we use the term strepto. So the words you see on this slide, single, diplo, tetrad, staphylo, and strepto, those are the terms for arrangement. But normally, like in everyday micro conversations, we normally describe the morphology and arrangement when we're talking about an organism. And so when we do that, we merge the two. So for instance, if I looked through the microscope and I saw pairs of circular cells, I would say those are diplococci. Right? So cocci because it's plural, we're not just talking about one cell. Diplo because they're in pairs. So you actually say the arrangement first and then the morphology. So diplococci. What if we looked through and we saw a chain of rod-shaped cells? Well, chain would be strepto, rod-shaped would be bacillus, or bacilli because it's plural. So we would say we're looking at streptobacilli. Okay? Now, if you look at this slide, notice that you do not see any bacillus cells for tetrad or staphylo. And that's because bacillus cells cannot divide along their long end. Okay, that means they cannot divide and stack on top of each other. Bacillus cells can only kind of link together end to end. So you will never have staphylobacillus, you will never have tetrabacillus. Okay. All right, so let's go ahead and practice on our own. I recommend you pause the video and describe the morphology and arrangement of these cells. Now remember, we normally just use one word to describe both. So you put the arrangement part first and then the morphology. All right, so let's look at the left-hand picture. So you first want to kind of try and find a cell all by itself. to look at the morphology or shape. So I'm going to look just right over here. It's not really by itself, but you can clearly see that it is a round cell. So round would be coccus. And because these are all arranged in big clusters, big groups, we're going to use Staphylo. So the answer is Staphylococcus. And if you say Staphylococci, that is fine as well. I really don't count off for plural versus singular. It kind of, again, just depends on how you're using it in a sentence. But if you know it's Staphylo and you know that it's cocci cells, that's what I'm really worried about. All right, let's look at this right-hand picture. So notice first the morphology. So let's see, here's one by itself, right? So that's not very circular. It's also not a spiral. So by default, we know the morphology is bacillus. Now to tell the arrangement, you want to look at what's happening the majority of the time, okay? So if you look, most of these cells are all by themselves, right? So we would describe the morphology and arrangement of this group of cells as single bacillus. All right, so that's going to be the end of this video. Next up, we are going to talk about the plasma membrane. Let me know if y'all have any questions.